This is another laboratory video. This is to go over the cell parts as well as cell division or mitosis. Right? So this coincides with exercise 4 in the lab book. If you want to follow along, you should utilize the lab book. And you can look on page 42 in exercise 4 for the diagram of the cell. So I'm going to be using a model to go through this, and this goes along with the lecture information that we've gone through. Right? So this is just a model of a cell. Right? Now this is a generalized cell. Different cells are going to have different specifics, and we'll look at some examples of that under the microscope. I'll actually have some video recordings of some microscope work. But this would be your generalized cell, just to learn the basics of the different parts of the cell. The first part of the cell that's important to talk about, and this is covered in the lecture as well, is the plasma membrane. So in the lecture material, I go in depth into the plasma membrane, the function of the plasma membrane, and the proteins in the plasma membrane, and what they do in terms of how they can move things into and out of the cell. That's going to be extremely important because how cells function in terms of their metabolism and what they can do is really dependent on how well they can bring things into and get things out of the cell. So you really want to pay attention to the plasma membrane portion. Now some of the other organelles that we would find within the cell, if we take a look, I'm going to bring this up a little bit closer. This main big organ here, this is the nucleus. The nucleus is referred to as the control center of the cell sometimes, and the reason for it is it's chock full of what's shown here is this kind of fibrous stuff, okay, these little fibers. That is chromatin. Chromatin is a form of stored DNA. And DNA is the genetics of the cell. It encodes all of the information to tell the cell what to do. So that's why the nucleus is referred to as the control center. Within the nucleus is where the DNA will be transcribed into RNA, and then from here it will be taken out and it will move to these organelles over here that are shown as blue with little red dots on them. Okay, that is the endoplasmic reticulum. This is specifically the rough endoplasmic reticulum. Those little red dots that are shown here and also kind of floating around throughout the cytoplasm, those little red dots are called ribosomes. Ribosomes are little machines, basically, that help make protein through a process called translation. They take RNA, messenger RNA that was transcribed from DNA in the nucleus. That messenger RNA comes to the rough endoplasmic reticulum where it is translated into protein. Proteins then really do all of the work within the cell. Right? They're both structural and they're also functional. Now you may also notice that we have some areas here that have that blue membranous structure, but they don't have those little red dots on them. Okay? This area here, these are smooth endoplasmic reticulum. And smooth endoplasmic reticulum, besides lacking ribosomes, right, they obviously are not going to be involved then in protein production because protein production requires ribosomes. So depending on where we see this in the body, what type of cell, this smooth endoplasmic reticulum is going to have very different functions. This will be quite important when we talk about muscles and how muscles work. We'll see that this specialized smooth endoplasmic reticulum is going to be important in muscle contraction. Now dispersed throughout the cell, we also see these orange structures, and if you look closely, they have a layer within them. These are mitochondria. Mitochondria are referred to as the powerhouses of the cell. These mitochondria are where cellular respiration occurs, and these mitochondria are considered the powerhouses of the cell, and this is where we have things producing ATP. ATP, or adenosine triphosphate, is a very quickly usable energy source for cells. Now in this region over here, we see this structure. This structure is representing what's called the Golgi apparatus. The Golgi apparatus actually works along with the rough endoplasmic reticulum, so the proteins are produced via translation in the rough ER. They're then packaged and sent to the Golgi. And in the Golgi apparatus, they would be modified and packaged for transport either out of the cell or elsewhere within the cell. Now the final structure I want to show you on this model are these paired structures right here. Okay? These are called centrioles. These are going to be very important in the process of cell division, which is called mitosis. Mitosis is the process of dividing the genetic material equally and creating two new cells. And we're going to look at that now. So when we think about cells, cells are really the basis of all life as we know it. So in order for reproduction to occur either on the organismal or the cellular level, cells have to be able to divide. In order for a cell to divide, it has to be able to divide its DNA exactly. The reason for that is the DNA is what has all the information to allow the cell to do what it's supposed to do. So if you're missing any of the DNA, you run into problems. So one of the first steps, if you take a look in the lecture notes, I go in depth into the cell cycle. Now the cell cycle is going to be an overview of what happens in the cell in terms of when. One of the key things that happens during the cell cycle is something called S phase. And S phase is the synthesis phase, and this is when DNA replication occurs. And I covered DNA replication in the lecture information. DNA replication is the process by which all of the DNA within a cell is copied exactly. Then later on, we have what's called mitosis. Mitosis is the process by which a cell takes that newly copied DNA and divides it equally into two new daughter cells. So mitosis is covered in the lab book in exercise 4, starting on page 46, where you see both photomicrographs and diagrams of the process of mitosis. And I'm just going to quickly run through and show you this in a three-dimensional model form. Okay. So here's our first model. Okay. In this model, this is a general cell. We've removed most of the other organelles, so we're not getting those in the way. What you do see is on this side, you do see it. We have two centrioles now that are starting to move away from one another, and they're creating this little starburst pattern that's called an aster. Right. If you take a look, the chromatin is starting to become condensed, but we still have it housed within a nucleus that has a nuclear envelope or nuclear membrane. Right. One of the first things that occur during prophase, or the first part of mitosis, is that this nuclear envelope starts to break down, right? And we're going to lose that nuclear envelope. And also this thing right here, which is the nucleolus, will basically be broken down as well. So from prophase, this would be early prophase. Okay. Then later in prophase, we start to see the centrioles are moving away from one another. The nucleolus is gone, telling us that we are definitely in prophase now. Right. The chromatin has become condensed into chromosomes, which are visible condensed areas of chromatin. Right. And we're starting to see this separation occur of the centrioles. Now later in prophase, what we have is the centrioles have now moved to the two opposite poles of the cell. The nuclear envelope is gone, so the chromosomes, the condensed chromatin now in the form of chromosomes, is no longer visible. The chromosomes have also hooked on to these fibers called spindle fibers that are produced by the centrioles. Okay. What will happen then is those spindle fibers will start to kind of push and pull and push and pull, and they're going to rearrange those chromosomes. And as we move into metaphase, we'll see that the chromosomes are going to line up along the middle of this cell. So we have one centriole on one side, one centriole on the other side, the spindles connect them, and the chromosomes are lined up. And you can see this would be a three-dimensional lining, right, where the chromosomes are going to arrange. So when we talk about metaphase, the chromosomes are lined up in the middle. The next phase then is anaphase. And in anaphase, we'll see that the two chromosomes that are copies of one another produced in S phase are going to separate. So we're now separating what were called the sister chromatids into two new chromosomes. And these are going to start moving away from one another as they're pulled on the spindle fiber. So this is the first part of anaphase. As anaphase proceeds, we start to see these chromosomes are now very separate from one another. They are being pulled to opposite poles. We also start to see the process of cytokinesis beginning. Mitosis specifically talks about the separation of the genetic material, the splitting of the cell and the rest of its components, the cytoplasm and the organelles, which aren't shown here. That's called cytokinesis, and it actually occurs when you get what's called a cleavage furrow, or a pinching of the cell. Now, as we move into the final phase, which is telophase, right, we start to see the nuclear envelope is going to reform. We have a much more pronounced pinching, right, or much more pronounced cleavage furrow, right, leading us into telophase, where we're getting two new cells, until finally what we have as a result are two now smaller daughter cells, each one with its genetic information housed in the form of chromatin, right, within these nuclear envelope, right? So here we have a nucleus. This would be a new daughter cell and a new daughter cell. Each of these cells, though smaller, they now have all of the same information that we had in our initial cell. Okay? So we've gone from a single cell to 2 cells. Now the cell cycle will continue, and these smaller cells will grow and eventually will undergo mitosis themselves. Now you should also look for separate videos that I will be posting that will be overviews of microscopy techniques looking at these cells under the microscope.